Good morning. Happy to kick it off for today. I'm going to tell you about the rollout of QBDVision at Viralgen, and we build a control strategy for our platform process. And I'm going to show you some of the things that we have done in the last year. Here's some disclosures, just so you have seen it. Just to walk you through the agenda for today, I'm going to introduce you to Viralgen, what they do, their manufacturing platform, their digitalization roadmap, and part of that, also the rollout of QBDVision, how we build a platform control strategy in there, use that for scoping process characterization, and automated a lot of documentation on the way that we needed. So the story of Viralgen. Viralgen was founded in 2017 by a joint venture of Asclepios Bioscience from North Carolina and Columbus Ventures from Spain. They were acquired by Bayer in 2020, and Viralgen is located in San Sebastian in Spain. As you can see here, also on the right, they have two facilities, one clinical facility you can see on the upper picture, which has 4 different suites for manufacturing up to 500-liter scale, where then the commercial building on the lower side has 9 suites with 3 independent modules to manufacture up to 2,000-liter scale. The Viralgen platform is based on the Proton cell line, which comes from Asclepios Bioscience. It's a standard triple transfection manufacturing process for the upstream part, and in the downstream, it's a standard downstream processing with ultracentrifugation at some point. Viralgen is working on advancing in that field towards perfusion processes in the upstream part and also getting rid of the ultracentrifugation, and it's soon to come. But most of the batches that we have manufactured up until today, and it's more than 750 batches, many of them at relevant scale today, we're more than 750 batches. We also many offer of them at relevant all the different AAV serotypes. With this platform, we can fit in our platform, may there be heparin binders or non-heparin binders. Looking at the digital strategy of Viralgen, we have multiple tools that we are rolling out, like an ELN solution, PIMS, LIMS, PI, SAP, and others. And one essential piece that we are also rolling out is the risk management tool, and we see QBDVision at that risk management tool that is mainly used by process development and the MSAT group. And today I'm going to show you more around the tasks of MSAT for process characterization and building a control strategy. Before we can even do that, and we heard that a lot in the last days, is that you have to start with structuring your data. So we aligned our terminology first in a way that we have defined all the terms that we want to use as a company-wide standard for manufacturing. That enables us to have FAIR principles in place, like findable, accessible, interoperable, reusability. And then all the documents can speak the same language in the end, right? This is all a searchable platform that we host on Power BI that helps us also to give access to the whole company to find the terms and use them. The process detail is also something that is really important. If you think of, like, if I want to process a transfer a process from A to B, what is the detail that I actually need? Like, how many parameters do I actually need? What materials do I actually need to include into that? And if you can make that a fixed set of information, this is the package that you can put from A to B. And this is something that we worked on to make transfers also more efficient up front. So how do we see QBDVision, right? In the end, for us, this is a tool that can structure our data. It's Viralgen's knowledge base for manufacturing processes with quality at source. So what's quality at source? It's a lean manufacturing concept, actually, where you say, okay, information, the earlier the quality comes in, basically the better, right? So here is like in every step of the manufacturing process, you want to get quality information and not at the end. But then it's exactly where we're going, right? If you have the information with a high quality at the very beginning, you can use it further downstream easily. Okay. So what we bring in is, of course, the harmonized terminology, the process detail alignment, as I just explained to you. We use it as a knowledge management platform. We bring in the experience from batch history, process version, process information from scales and sites, so it's all in one solution. When we look at the rollout plan for the last year or for this year, we started building a control strategy. We applied multiple development MSAT tasks in QBDVision, and then we used this information that we put in to get structured information out in automated documents. I highlighted that a little bit with C, T, and A so that you can find it in the next slides whenever I'm showing how we did it. So we start with our risk strategy. Usually for all the quality by design tasks, you start with your quality target product profile. And what you want to do is here define high-level targets based on your best practices that we have in place at Viralgen, so that we can assist our clients defining the QTPP for their product based on non-clinical or clinical data. What we can do in QBDVision, though, is then automate the document to ensure that we have consistent naming, data integrity, data quality for all the downstream applications of the same information. Here you can see a doc builder template that we built. So then, and on the right is basically the Word document that we can generate out and share with our clients with a very high standard for every client that comes in. The next piece is normally the CQA risk assessment. Here we took a structured approach. We screened more than 4,500 papers together with Bayer in the AAV space to make sure that we can cover all the serotype information and so on. Then screened abstracts at Viralgen, boiled it down to, in the end, about 60 citations that we use to justify why a CQA is critical or not, on top of pharmacopoeia information and so on. In the end, it enabled us again to have a serotype-specific CQA risk assessment available, built in Doc Builder as a template that we can then just easily export and adjust for every our client's needs. The next piece is then applying process descriptions or transfer gap assessments in the software. Here we want to enable Viralgen for rapid process description for all of our processes so that we can dynamically generate information like parameters, materials, equipment, disposables, in-process controls, whatever we need, when we need it, in the format that we want. We have the same namings like in all the other documents, right? So the standard comes the second you fed in all this information. Again, we can generate block flow diagrams, use the doc builder to automate the documentation around that, and we're going to see a little bit more on the next slides. What we also want to do is align our scales. So we have multiple scales that we're using for tox until commercial manufacturing between 50 and 2,000 liter scale. And we want to have all these scales aligned in the software with the same package of parameters that I described at the beginning, to then use the same templates for all these scales. Also, we want to have version control in the software so that we can version our platform and also the processes for our customers more easily. Looking at a process description example, so here this is a document that we generated. For example, bring in sub-information from equipment, from materials, disposables here. Also, if we want to look at process parameters, you have information about the criticality of these parameters. You can use description fields to help write batch records in the end. You can use the display functions of QBDVision to show the structure of the process in block flow diagrams on the right here. So these are the kind of documents that we are generating right now. Okay, and the next piece that you want to do is the process risk assessment. Once the process is in there, basically based on a transfer or based on a process description, whatever it may be, you can start with the risk assessment of the process. And you can, for example, then connect the CQAs to the process parameters and the material attributes. Here in this example, we are looking at the column unit operation. The bed height of this column can be between 18 and 22 centimeters. So this is my failure mode. With two normal operating ranges considered, and we're defining the failure effects, and we do that in a big team of experts for the whole process. And once you have done that, you can again automate this with Doc Builder to generate these huge documents, these huge risk assessments. And for those of you who have done that by themselves before, I mean, these are documents that are really error prone. Right? It's very, very, I mean, maybe 200, 300 lines, right? And it's very easy to miss something, to make small mistakes that in the end lower the quality of this information. And if you generate this with QBDVision, for me, it was a huge improvement of the quality and the speed that we can generate these kind of documents. The next piece that you can do then is scope process characterization with that, right? QBDVision has this built-in cause and effect matrix that you can use where you plot your CQAs against your process parameters. You consider your known interactions, the platform knowledge, literature, to then combine this information to help you to scope the process characterization. You can, for example, use the risk numbers as a priority label. Basically, you can see how many CQAs are linked to one parameter to get an idea how important that parameter is to be investigated in process characterization, so you can rank them. You have the edges from the CDMOs that help you, and then you can include the right parameters into your design of experiment approaches or one factor at a time experiments based on this matrix. For example, here you can see there's 4 parameters on the upper side that are impacted by the same CQAs. So it might make sense to include all these 4 in one DOE experiment. So this view just helps you to make better decisions for your experiments. Because in the end, everything is connected in QBDVision, right? So by doing the risk assessments, you get these connections on the side. Here I'm showing a bioburden CQA, and then here I can see inside the software, where am I actually controlling bioburden in my process? Like, where's my in-process control? And then I can also zoom in here to see which parameters are actually impacting the risk of bioburden in my process. So this gives me a very good overview of my control strategy that I have in place for this particular CQA. And I can use that also to train people to handle deviations, to see basically what's the capability of my process. As an overview, we have just linked all these standard tasks that you do in late-stage projects. We put that into a structured data set, and now is basically the point where I can benefit from my quality at source or quality at the very beginning in late-stage applications, right? We've seen it yesterday. You can use it for batch to MML exports in MES system. Very nice demos yesterday. You can think of, if you don't have MES systems, you can automate batch records. We've tried that. It works. It's super nice. I mean, you can automatically update based on a transfer a batch record template in DocBuilder. Super nice. Another feature, and that's something we're going to show in Sue's conference next week, is also to automate submission documents. Because in the end, they want to, what you want to do is show a certain piece of your manufacturing process in a CMC submission part, but maybe not everything, right? So DocBuilder is also made to show explicitly some points of this. And it's a really nice stuff that we also tried. Okay, and so in summary, QBDVision allows us to compile all information in a single platform with quality at source. All documents are aligned. They speak the same language, the same terms, the same quality. Everyone can work simultaneously on these documents. Everyone can find them, no silos. The changes are tracked and updated in these documents if you do it manually. We can easily build a control strategy, use that to scope process characterization with significant time savings and also a way higher quality of these documents for our customers. If I look at the main potentials that we see in the software, for us it's basically integrity. Yeah? If you think about you have to change one parameter of your manufacturing process because, I don't know, you have to update it based on a deviation, let's say. And you have to go to 10 different documents to update this. The likelihood that these documents run out of sync because of small mistakes is really high. On top of that, the manual effort you have to do going into each and every document and change it, and then again get them approved, is also super high. So that's something where we see a lot of benefit if I change the value only in one location and it updates automatically in all the other. That's the benefit, right? Integrity, fast. So we see time saving by automating the documentation for up to 90%, I think, and we can really focus on the content, not on the creation of the document anymore. We also have the vision that we can use a copy of the platform for every new molecule coming in as a starting point, and then evolve basically this platform based on the needs of the customer the specific changes that we have to apply for the product. It's our knowledge management tool. QBDVision enables us to build and maintain a manufacturing platform with all the relevant information, historical data from clients, in a very efficient way. Of course, there's also challenges. I want to talk about that. We heard around, a lot around change management, and I want to give you a little bit of the perspective from the rollout, right? So for me, what I've learned in the last couple of years, that it's very important to enable the rollout team with the appropriate level of tasks. So if you give them too much, they're overburdened. They're going to skip, right? But if you find like the right tasks and then slowly ramp up, they will fly at some point, right? That's what I saw in the last year. Also, as a rollout manager, as the expert, so to say, you also have to let go. Yeah, that's also something that I actively have to do. It's very easy to do it by yourself because you're the fastest and you know how it works, but you really have to step down and let the other people do it so that they can actually train themselves in it. Also, complexity is a function of the detail you want, and that means basically think about your use case in the software and don't put everything, right? So put whatever you need for the task you want to accomplish and then build processes around it, right? So you need processes. For example, how you version a process, how to reuse a platform process, how to make it project specific, right? If you don't have this in place, it can get as messy as a Word document, right? Then, of course, you have this standard competition with Excel sheets, right? They're very simple. We heard that multiple times, but this is definitely always a case. If I want to look in the future, like the stuff in gray I'm showing here, you have already seen. I've demoed this, but we think of QBDVision as a validated system in the future where we can then generate automated batch records in. For example, we can do transfers, versioning in. We want to hook it up with the API function to our PIMS, LIMS, and subsystem. With the validated information in the future, it's also open for batch 2 MML exports, as we've seen yesterday. We want to leverage that as well. And of course, also the CMC sections for our submissions can be automated. And with that, I thank you for your attention and take your questions then after the session. Thank you.
Hello, everybody. So today, I just want to talk to you about a case study my group and I worked on, wherein we leveraged the QBDVision API to better integrate the QBD system with our internal workflows. So to just get right into it, Sanofi, as I'm sure you know, is a very big company. In 2021, they started the mRNA Center of Excellence, or COE, dedicated to the development of mRNA vaccines. It has a few hundred employees across France and Waltham in the United States, where I am based. And I work under John McGuire, who you may remember from last year's conference and his talk. I myself, as a data manager, really focus on all things related to our process and product data, including ingestion workflows and managing the data model. And although he was only with us for 2 months, our summer intern this year, Jacob Kalka, was essential in bringing this project across the finish line. Our group mission, our driving force, is to get data into the hands of the people who need it as efficiently as possible. And the first step in that process is data capture. During this conference, we've heard a lot about how PDFs are spooky, and they admittedly are. It would be ideal if our raw material vendors, our CROs, our CMOs handed us structured data tables that we could run forward with. Unfortunately, in my experience, they are not only not willing to do that, but do not want us anywhere near their databases. So what that leaves us with is our entire process completely haunted by PDFs and PDF usage at each stage in the process. What I really want to focus on for this presentation is just one of these documents, which is the raw material COA. Each instance of a new raw material is assigned a unique lot number when it's generated, and it is tested to make sure that its material attributes fall within the expected specified ranges. The summary of these analytics is what gives you your raw material COA. When you produce a product, you have to use many different raw materials across many vendors, and each of these vendors has their own formatting for the COA, their own manner in which they generate lot numbers, and their own system for actually naming the PDFs themselves. What this ultimately results in is when you produce a product, at the end of the process, you're left with this giant pile of PDFs that's not very helpful. And in order to actually access the data held within, you need to do some amount of processing to that data. So we take the COAs one at a time. We extract the data into a structured data table. Shows up better on the big slide. Validate that structured data, and then ultimately file it away in a searchable document library. So this is the idea. Our initial process workflow to meet this need looks as follows. And this is very complicated and all over the place, but in brief, worker A goes in, uploads the COA into the QBDVision document library, then goes into an Excel workbook to enter the material attribute data. Worker B goes and then validates that data, and then worker C, a quality assurance employee, goes in and saves over the public version of this workbook to update the live version of this database. So a few things. This is a very manual process. You can see that of the 27 steps listed, only 3 of them are automated. And this requires coordination across 3 different employees, one of them in a totally different department. In addition, this database is not continuously updated. Instead, updates are done ad hoc through version updates, and every version update generates 3 additional documents that themselves need to be managed, which you can see highlighted in yellow. So what we want to do here, we set out, is to optimize this process because we saw there's a lot of room for improvement. The first thing we really wanted to look at is replacing Excel. Now, 21 CFR Part 11 is just a section in the FDA's code for regulations that outlines the criteria for electronic records and electronic signatures. It ensures that you have data integrity, security, and reliable data, which when you're dealing with GMP data is especially important because it can affect the lives of real-life patients. Excel is not compliant, but if you add enough business process controls, you can kind of force it into being part of a larger compliant system. In practice, what this means, though, is that you end up with a lot of additional manual steps, as we saw before, and a lot of additional documentation that's generated that themselves need to be managed. Alternatively, if we use QBDVision, since it is compliant, we don't have to deal with any of these workarounds, and we can just move forward. And the material library within QBDVision actually suits our needs for our raw material COAs really well. So for each material, there's a structured table that contains the data for the attribute specifications, as well as a structure table for the material lots. Every time a new C of A is uploaded into QBDVision, the text is automatically extracted from the PDF as a first pass on data entry. There's some color coding to pinpoint where there's areas of less certainty as part of that extraction process. And then ultimately, the uploader has a chance to make edits to what was extracted so that it aligns with what's actually there. Once uploaded, the actual material lot information includes a structured data table of all of that material lot information, as well as a link to a copy of the COA, the PDF itself that was used to upload that information. And this is all fantastic, but for our purposes, not exactly what we're looking for if we can't get the data out, which brings us to the QBDVision API. An API, or Application Programming Interface, is a software intermediary that facilitates the communication between two applications. And it's commonly compared to a waiter at a restaurant where you put in your order, it goes to the kitchen, and then it brings back your food. For our purposes, this is a way in which we can access the data within QBDVision within another application. And so as part of the request for one of these APIs calls, there's three main parts. The first is the API key, which just gives us access to the server data. The second is the URL, which points us to where that data is. And the third are parameters, which are really part of that URL that help add some additional level of specificity to some of the data that we're pulling out. In terms of the response, there's two main pieces, one being the response code, letting us know if they received our order, and response headers that contain the actual data itself. Now, through combing through the QBDVision API documentation, some trial and error, and some help going back and forth with Luke, we were able to land with our URLs. The first of which, very straightforward, the material library URL, which just returns us a table of all of our materials, as well as the material level attributes. In order to get the attributes at the lot level, we use a material information URL, but that can only be called for one material ID at a time. So we need to get all of the material IDs first, and then dynamically assemble these URLs and dynamically call, make these API calls after they are assembled, along with their appropriate parameters. So when all of this comes together, we end up with our aggregate URL that we can use to get to our lot level attribute data. The low-code environment that we used to both assemble these URLs themselves, as well as to make the API calls, is called dataiku. And dataiku was also used to then parse the JSON data that was returned from these API calls, clean them up, and combine them into our final data set. An added benefit of using dataiku is that we were able to add in some automation because we're able to do some simple triggering. The most straightforward of these is just periodically calling a workflow. Dataiku is another function wherein if you call a workflow and it meets a certain criteria, you can then trigger an email to be sent, which is relevant to us because an email being sent can trigger another low-code environment, which is Microsoft Power Automate. Taken together, we can trigger Microsoft Automate flows whenever QBDVision updates its data. Taking this all together, we're able to land at a much more simplified, streamlined flow. So we receive an email to our group inbox with an attached COA that is downloaded into a SharePoint folder, which triggers a new list item to be created for a SharePoint list, which is just there to track metadata on that document. This then updates our task board, which directs our worker A to actually upload the PDF to QBDVision. Dataiku pulls that data out from QBDVision and uses that to update the metadata on the SharePoint list, which updates the task board and compels worker B to verify the data. That verified data is then used to make up our final data set and can be used to power a dashboard or any further processing that you want to do with that data set. Now, this is a lot, but the main takeaway here is there's really just two manual processes in this workflow. Everything else is automated, which is the uploading to QBDVision and the verification of data, in part because they kind of have to still be there if we're still getting this from PDFs. The other nice thing is we've completely eliminated the necessity for the QA role, so they can have some time back to do more important matters. We also added in some process monitoring windows to keep tabs on this overall process. The first is very straightforward, is just file dialog window of the file folder that the COAs are dumped into. Flow watch is this bird's eye view to just give us a timestamp of when our automated flows are running. Task watch is a Microsoft task board that gives us a snapshot of when or where these various approvals and data entry tasks are for all of our documents. DocWatch is here to give contextual metadata to the PDF documents. And then the Dataiku export is synced to data set within Dataiku, and it really is there to pull in the data that's pulled in ultimately from QBDVision. Power Apps can also then be used to aggregate all these windows into a single view, which just helps when monitoring the process and makes it a little bit more convenient. So real quick process walkthrough. In terms of monitoring the process, it really makes sense to break things down into 3 milestones that break up the cascades of automation. So there's first the email that comes in that triggers the workflow. There's the cascade of automation that follows that. There's the actual entering of the data and uploading to QBDVision and the cascade of automation that follows that. And then finally, the verification and the subsequent cascade. So in this case, we have received the email and we get all the way over to updating the task board, which results in what you see here. So we have a new PDF in our COA inbox. We have a new SharePoint list item to track the metadata for that document, and we have a new task in our task board for data entry. After this PDF is uploaded to QBDVision, the next cascade occurs, and we get new data in the Dataiku export, which is used to update the metadata in DocWatch. And there's a new task to approve this information in the task board. One note here is that because you can draft data entry and not yet submit it within QBDVision, we have this sort of in-between state, which is shown by the half moon on the draft task. So that is not actually marked off as complete here. It's marked off as half complete. And if you look at the QBDVision status in DocWatch, it's still as draft. When that gets updated to proposed, then the draft task is marked off as complete. Then lastly, after we verify the data, we've got our final cascade, which shows that the document within DocWatch is shown as being fully approved. Our approval task is fully marked off, and all of the approved parameters within DocWatch are then used to update the original PDF and rename it with a structured name using all of the verified parameters from DocWatch. So, in summary, we were able to use the QBDVision API to get all the benefits of its secure, compliant database without having to sacrifice anything in terms of accessibility. By using Dataiku and Power Automate, we were able to get additional gains in automation, and as a fun bonus, Windows for process monitoring. For our next steps, we really want to expand our use of the QBDVision API and pull all of the data that we've got in there out. And we also want to expand the scope of our document process monitoring in general so that we are monitoring the data entry status for not just our COAs, at least not just our raw material COAs, but our product COAs, potentially batch records, et cetera. So, thank you.
Hi, everybody. Grant Henderson, VernalBio. I'm going to talk a little bit today about called a case study where we've implemented QBDVision, and really just QBDVision, no integrations, no automation, to facilitate our platform tech transfer operations. I'm going to start off, tell you a little bit about some of the challenges with the CDMO especially, and then how we've used QBDVision to map out a platform for our manufacturing operations, plural, at various life cycle stages of the product, in a sort of a generic way that can be applicable to really any product that might come through our doors. And then I'll end with a very short case study where we've done this with one of our own catalog products. So, who are we, what do we do, and why is that important for today? As I mentioned, we're a CDMO. We don't know the product that we're going to manufacture before we sign a PO, or before a PO is signed on our behalf. And so that means that all of our manufacturing processes have to be sort of platformed. We put months of effort into developing those platform processes, not knowing what the actual RNA sequence is going to be that gets dropped into it. And then when the customer comes in, we learn that sequence, we put it into the process, and we make them their RNA product. All we really know is that that product has to be high quality at the end. If it's a drug substance that we're manufacturing, there's certain sort of industry-wide standards for that. If it's an LNP RNA, there's also kind of standards for that. And so, we can go into various literature, whatever's out there, and try to understand what that's going to be. But at the end of the day, if someone comes up to us with, say, a vaccine with a dose of, say, 30 micrograms that they get once per year, that's very different than if it's a therapeutic, where they're going to get, maybe 10 times that over the course of, with multiple injections over, the course of months. And so you can imagine in those, especially things like the impurity profiles would be different. And so you have to account for that when you're trying to anticipate what your QTPP might look like. So the reason I bring this up is what it means is that when you have a standard QBD approach and you have to start with product knowledge and then in order to progress through process knowledge, that kind of gets flipped upside down, right, in our use case. So what we have to do is generalize, just essentially learn everything we can about RNA, which we've done, and come up with sort of a one-size-fits-all specification, all right? That can be super comprehensive. Not every test is going to apply to every product, but it's a lot easier to remove than it is to add on a project timeline. So that's what we do. That's who we are. And those are the challenges that we face as a CDMO in the RNA manufacturing industry. So I think we've seen various variations of this slide. I won't take long on it. I'll just draw your attention to the bottom, right? So the way we've organized our manufacturing. We have your standard product life cycle, which we've broken up into 3 stages for product manufacturing purposes. We provide drug discovery or research use material. We provide material that can be used for pivotal tox studies, GLP tox. And then we have our phase-appropriate GMP manufacturing. Those give us 3 big buckets in order to do process development. The process that you run to make product for any of those 3 big buckets, those processes will look different because the requirements on those products are different. Different control standards and quality standards. Also very different batch sizes and throughput requirements, right? And that's sort of summarized here. Again, focusing at the bottom, the process. So when we're doing drug discovery, it's generally a high throughput approach. You've got a customer that comes in, they want 100 different molecules, and they want 1 milligram of each, right? It's a standardized process. You can go into the shop floor, you can run the exact same manufacturing process to get all 100 of those molecules, and if 88 of them, 90 of them turn out great, in a lot of cases, that's good enough, right? And then you go back and you say, our standard process, didn't work for these 10, right? Should we do some extra work here? But in general, you're running a standardized approach for those. The scale is small, maybe up to 500 milligrams, and it is an open process, right? So this is, not going to be suitable for applications that you would have in later phase. When we go to tox manufacturing, right? So something that would be for a GLP tox study. Now we tighten up those controls, right? We also need more product. And so now we're getting into our single process, our single product manufacturing process, not necessarily running it at scale, but a process that is scalable, and a process that might take adaptations in order to get it to work perfectly for this molecule. And those adaptations are justified because of where it is in its life cycle. You're willing to put the effort in to improve efficiency and ensure that you'll be able to scale this into your commercial phase. Process closure, whereas we had previously an open process, now we're going to do a risk-based process closure, where we're going to try to limit things like endotoxin and bioburden. And we'll do that through risk assessment. And then you move finally into the GMP. I'm not going to try to cite GMPs, but it's, there are some, that's when you're in your fully kind of finalized state of the process. Right? A little bit about the process, just for some context here. As I mentioned, we do this 2 different ways, at least 2 different ways, really. When we're doing drug discovery, you'll see all the equipment on the top, very much what you might find in an academic setting. A lot of manual steps, some automation, but even the automation that we do have is kind of on an islands of control basis. People are walking up to a machine, turning it on, running their operation. The data does get offloaded to the cloud, but it's just going to a folder where they can then receive it. No different than walking up with a thumb drive. It's not getting shuttled around and adapted into organized data sets. As I mentioned, we have, I'm going to focus on the 2 operations today for plasmid, which is a template for RNA production. via an IVT reaction, and then the RNA itself. I'm not going to get into the drug product manufacturing. I figured this would be enough. But just at a really high level, in order to make an RNA drug substance, you do have to prepare a template. That template is then used in an in vitro transcription reaction, which is the same way that RNA is transcribed in your body with an RNA polymerase. Then there's a downstream operation to purify it, right? So RNA manufacturing in general, you could probably think about it just as a series of upstream and downstream reactions. Upstream to make a material, downstream to purify it, and then you repeat. So, in this slide, I guess the take homes from this is it's a complex process. Last count, I think we were up to 14 unit operations to go all the way from cell bank to drug substance. So that's a lot of process knowledge management, because every one of those unit operations has materials, has process parameters, has material attributes associated with those materials, right? Everything that QBDVision does very well in keeping track of that. So we've used QBDVision for that purpose, right? The way that we do this for platform, right? Because what you want to do is you want to set this up once, and then you want to copy it every single time you get a new customer in. So the way that we've done that is, as I mentioned, we create this model product. We come up with all the attributes that we think might be important for an RNA medicine, and we put that into kind of a mock TPP. And from that mock TPP, we have a mock QTPP, and we finally get down to finished product attributes. So we have this sort of model imaginary product that we think could be, have everything that any real product might have and then some. And then we do the same thing for the GMP process, right? So we do a lot process development in sort of the traditional sense, where you identify your unit operations, go in, do the studies you need to parameterize those, and then that is digitalized in QBDVision for the GMP scale, right? The trick here that we figured out was that once you have that GMP scale process, right, what we want to know is for any of those targets that our customers might have identified through the drug discovery phase, how confident can we be that when we scale up to GMP, we're going to have the same product quality that they had when they did their drug discovery, right? And so what we did is we mapped that GMP process back against the drug discovery manufacturing process. QBDVision made that pretty easy because all we had to do was copy the process. So we had our one project that was the platform. And so we could just take that process and copy it, and then go in that copy and adapt it. So where we may have had a 20-liter bioreactor to make our biomass for plasmid purification in the GMP process, now we take that and we relabel it, and now that is a shaking incubator with a 2-liter shake flask in it, right? So we can take that process, pull out the steps you don't need, adapt over maybe some of the steps that have changes, and now we have a drug discovery manufacturing operation that is mapped to a GMP manufacturing operation. And we can do that as many times as we want. So I said we had 3 buckets. You can also do that, say, for GLP tox material, right? And so the nice thing is that as you do this, all the changes that you're making are being tracked, or they're at least being characterized in such a way that they can be reported out on later, right? And that's what we do next, right? So there's this nice little tech transfer report function that you can run. You run that tech transfer report, and it'll tell you all the differences that you did. And if you set up your material attributes and your intermediate attributes and your finished product attributes and your process parameters all correctly, according to your risk management plan, not only will it tell you what the changes are, but it'll also output a level of risk associated with that change. This is the first step I was talking about, where we have to build our GMP process in QBDVision. The way we do that everyone probably will do this a little bit differently. The way we do it is we set up an outline, just list all the steps. Once we have all those steps and we know what that process ought to look like, we go back to our process development, foundational data. Although I think yesterday, foundational, the master data was what was described as foundational, and so this would be more like bedrock, maybe, because it's a little bit below. So we go back to that bedrock process dev data, as well as our equipment list, and then with all that information, we throw it back into QBDVision to get the full process model, right? The challenges that we encountered, so once we did that with our platform process, it did throw up some flags for us, which was good. It actually made it so it was worth doing. And what we could trace all that back to was the fact that commercial batches in the RNA space are relatively small. We're making hundreds of grams of material typically, as opposed to like a protein therapeutic where you're in kilograms. And so the equipment selection and the use of the equipment, in order to, what we found was that we were often trying to run our equipment at the very edge of its capability, right? And that we were able to flag that. It also, we also found that managing our material attributes for starting materials, right? Because we have not only life cycle management here, where we have, say, a material we want to make for a phase 1 clinical trial versus a tox batch versus a drug discovery. And so those all have different quality standards applied to them. But we also have, the plasmid starting material, for example, is a starting material. It's not a drug substance, it's not a drug product, right? And so you have to think, well, do I want to apply the same quality standard to that? And if you do, when you first set up your risk management plan, you have to take that into account, right? That you're going to be applying multiple quality standards and you have multiple, different severities of risk based on where you are in your manufacturing process. So that was a challenge for us. I won't go through all of them, but the big one that we found when we were transferring was that there are a lot of time steps. So we have 3 chemoenzymatic reactions. Each of those chemoenzymatic reactions is timed. And so the time of addition, as you scale up, becomes very important. And so mapping all that out in the process was a bit of a challenge just from a process engineering perspective. But it was actually relatively easy to track in the software. So we had all those sort of high or medium risk type of flags that went up. On the life cycle side, it was the stuff I was talking about where it was just, do we apply this quality standard? Do we apply that quality standard, right? But after we'd worked through all that, you get this tech transfer risk assessment. Once all the mitigations are in place, you get a whole bunch of blue squares and ones. At least in this particular one that I pulled out, those all indicate low risk. And I just decided to throw this up as an example of what the output looks like. This is for the initial cell culture volume. So it does this on a parameter by parameter basis. And it just shows that, in this case, we changed the name of the unit operation and we changed the volume of a shake flask, right? So, but all those changes are tracked and the risk associated with each one is also tracked. And you can choose to mitigate or not based on your level of risk acceptance. So how did we apply this to an actual product? We took that entire project that we had set up for the platform and we copied it. And once we had it for a specific product, then we could go back through that QTPP and actually make it specific for that one molecule. So we do that. And then because you have your, we had already done a research batch of this material, we could actually go through and put in the values from that batch, right? So if there were process parameters, we could go through and we could actually enter those in and say, this is how we actually ran it. And then so you do that tech transfer report. And because we had mitigated everything based on the platform, didn't really flag any major risks when we did it for our case study. And so we just decided, okay, let's make the batch, right? And these are the results. So what you're looking at right now are capillary gel electrophoresis data and HPLC data on plasmid that we made, as well as the RNA product that was transcribed from that plasmid. And so the top line is the 5 milligram batch that was the drug discovery material, or the drug discovery grade material. What you can see in the plasmid is there are actually 2 peaks there. One is supercoiled DNA and one is open circle DNA. In the linearized plasmid, you take that plasmid, you cut it, and then that's the actual template for RNA. That second peak that was open circle and the supercoiled, because they're the exact same molecule, just in different configurations, they collapse into a single peak once you linearize them, and you see that. And then after transcription, the final RNA is the large peak there on my left, your right. And so this is a very good, if you're not accustomed to looking at RNA QC results, this is a very good, high quality RNA. And so that's what we were trying to match. We scaled that up to 1 gram, okay? So a substantial scale up. And what it looks like to me is that we appropriately mitigated our risk through this process. Where the 1 gram batch, we see the exact same profile. This is HPLC instead of CGE because that's our QC test for that scale. You get your 2 peaks, one's supercoiled, one's open circle. That collapses down to a single peak with your when you linearize, and you get your single peak for your RNA with no errant peaks, no evidence of degradation. Overall, we look at this as a pretty successful outcome. To summarize, if I was trying to explain to a group how to do this, this is what I would tell them. I would say, sit down, figure out what the objectives are. Right? In our case, the objectives it was not a single drug product. We could not sit down and go through a full QBD workflow. And so we took that into account, right? We said, okay, what do we know? What don't we know? Where can we start? And for us, it was just, we knew we needed to have high quality RNA. We knew, we do apply this to the LNP also, not shown today, but we knew the attributes that we had to kind of optimize for LNP. And we sat down and we wrote those down and we captured that in our workflow. That was at least a step 2 where we generalized the attributes of our dosage form. And then we planned and we modeled the process at each scale, right? Starting with the larger scale, working backwards. That was key for us. I think if we had tried to create the small scale process, we would have had trouble doing the scale up because it would have been hard to create those linkages between operations that exist in the large scale but don't exist in the small scale, right? So by going backwards, we had all the operations and we were able to remove the ones that we didn't need. Run that tech transfer assessment, do any mitigation you need, and then you have your platform tech transfer kind of project. Use that, copy it over, adapt it as necessary for any given molecule, and then as you run your risk assessment there, if there's any mitigation left, perform it, and then go ahead and run your batch. And like I said, that worked for us in this case. So thank you. And I'll take questions with the rest of the group.
All right, any questions from the audience? Okay, one right here.
I think those are definitely 3 excellent applications of the QBDVision software. So thank you for that. I'm curious, as far as the process of, purchasing, implementing, using, did you have these use cases in mind when you first saw the software, or did it take, some playing around and kind of convincing of senior management the value there?
I can answer it at least for myself on that. So I've been at my current company at Sanofi for about a year now. And I had some understanding of QBDVision, but the whole reason why that initial flow was set up the way it was is because there really just wasn't the internal knowledge of APIs in general, and certainly not of the potential within QBDVision to do that connection. So there's a way in which upskilling in any of these things, but certainly with QBDVision, understanding a little bit kind of potentiates more and more. Like as we solved smaller problems, we saw like, oh, now that we've done this, we can leverage the data we're pulling with this API to do all these other things. But yeah, no, it was almost a happy accident that we stumbled into the API thing. And part of what I really want to communicate here is that we initially really thought of QBDVision more as a data silo, and we're not aware that it is so easy to get that data out of QBDVision. And like, I'm not a developer. Like, this is something that is very accessible to everyone.
I can say that our use of QBDVision, I think, has evolved with the software. You know, the tech transfer feature that I showed, I don't think was part of the software when we first started using it. And so, you know, when that became available, it was you know, we had already known that we wanted to map our GMP manufacturing, our drug discovery level manufacturing. We knew we wanted to map all of those. So when the tool came to compare them, like that was kind of a no-brainer. And the same is true for other features they built in, like the doc builder. You know, that wasn't there when we first started using, but we now use that for process descriptions. No, when we first started using it, it was really just as a process knowledge curation tool.
Yeah, I could also second that. I mean, when we started using it, the capabilities 3.5 years ago were not like it is today, of course. And it was a co-development sometimes, and a journey together where we build new features based on recommendations and so on, so that we get to these kind of use cases like transfers that we can actually build into the software. Speaking as a Bayer rollout manager, for Viralgen, I would say I already started with all the tasks in mind that we could generalize or automate and make their lives better, pitching it basically on the data integrity problem, and then that worked quite well as well. Yeah.
Thank you. Any other questions from, I thought I saw one behind you.
Over there.
I have one for Blake, I guess. So you said you moved the data from CFAs or whatever into QBDVision. Why QBDVision? Why not any of the other ELN system where QC is using?
Well, certainly not the ELN systems that we use.
ELN or LIMS, for that matter, yes.
QBDVision, in part, it was because we happened to be using it already. I think, we don't want to have like system bloat, so if we can have a system we're already leveraging flex into a new space, we absolutely welcome that as long as it is appropriate. So I totally can imagine if we started out with a similar system and went through the similar process, if they did have that kind of accessibility with the APIs, maybe we could be talking at a totally different conference.
So obviously, like your manufacturing or QC space will not be using QBDVision, or is it?
Well, so our QC space right now, they are using an internal solution, which is still under development, which I think is part of that. And this was really focused on, at least for right now, less the QC data and more what we're getting from our raw material vendors. But potentially we do want to, you know, flex into at least getting some of the release testing, characterization testing, since the data model is really similar. So I think it's like an easy win, you know.
Thank you. Grant, for you, there's another question from my side at least. You said about bedrock data, so you collected the bedrock data and you linked it with the manufacturing steps. Is that how you did it in QBDVision?
No.
Or manually in that space?
It's manual, right?
Yeah.
So we're still using, Excel
Okay.
for the most part to capture that and to do the actual process simulation, and capture of data. It works okay, right? Like there's still a manual step of saying, okay, this is based on my simulation, this is the, say, flow rate, right? And then transcribing that into QBDVision. The nice thing, though, about having the QBDVision is once the process engineer responsible for that step has put that in there, it becomes a reference for everyone else. And you don't have to be able to, go to the spreadsheet. And I don't know, maybe that's a crutch. I have been talking to folks about this a little bit within my group of, try to make the information you're keeping not just for you, but for the group. But at least this gives them an outlet so that there is like a reference point for when we need that information. It's there, and we're not scrambling. When we go to make a batch record, like if that guy's out that day, like it's in the spot where everyone knows to look for it. Yeah.
Okay. Joshua, for you maybe, you talked about let go piece. So through this process, how easy was it to train people in QBDVision? Your scientists, I guess.
Yep. Yeah, I think last year I showed also the user concept that we have behind that. So we have different user levels, right? There's super users that are supposed to know everything, and to train them, I think that's the most difficult part, because you're competing normally with other tasks that they have to accomplish. So you get maybe 20%, 30% of this FTE, right? So you really have to figure out, as I described a little bit, just scratching a little bit on the surface, right? That, if you give them too much, they're not going to accomplish it, or they're overburdened with that. So, it's really easy stuff. Like, go with Excel first, right? Like, give them a sheet to fill, right? And then they figure out, it's going to pop up in QBDVision once I upload it, and you do the upload with them, right? So, like, step by step, and at some point, they start only editing stuff in the software, right? And then you have checked the no Excel box already, right? So, and then you can continue there, like, let them experience the advantages, right? Like over the time, like, I don't know, the material library we've heard, very nice feature. We tried that a lot as well. We tried the QA stuff with our QC department as well. Try to bring in departments, like find out what's their need. For example, at Viralgen, we also talked to QC department, and we found out, okay, they have a lot of assays and they're in different statuses, and where can we track that? And what is all the documentation around that? So that was a big need for them, because it was all spread out. And we said, okay, we can make a report out of this, out of QBDVision, and we can bring all this information together in one place. And then at the same time, MSAT was benefiting from this for the CQA risk assessment, because suddenly all the methods were in there, right? So it's like building up a little bit, and then everyone gets what they need, and yeah.
So it is a slow adoption, or is it something that you factored in to make it slow, so that everybody can get into it?
It's definitely a slow option, definitely, right? It takes time, you need that time. It's change management. You cannot enforce change too quickly. You're going to get resistance. So I think depends also on the people. So how interested are they? How much time do they have? And that's what can define the pace.
Thank you.
I would add, if it really depends what you're comparing it to. Like, if the bar is Microsoft products, for example, right? Mm. Those are the software that we've all used for all our lives, right? And we've kind of gotten a sense of familiarity with it. So I don't think that's the right comparison. You know, having just recently onboarded EQMS, an ERP, a LIMS, right, an MES, the learning curve on QBDVision substantially less than any of those, right? So you're not in for that kind of an experience, but it's also not something. It's pretty close to pick it up and use it, but there is a little bit of a learning curve.
Thank you.
All right, I think that's all we have time for. Thank you all so much. Thank the three of you for joining us.
All right.